We have four speakers today, four great speakers. Professor Lilian Chapman in biology, Professor Lilian Childress in physics, Professor Brendan Johns in psychology, and last but not least, Ms. Odessa Grimald from the Department of Psychology. So we're going to try something new this time, this Sun and Science for the summer. Following the presentations, you, the students, will have the opportunity to answer a skill testing question about each presentation. And the top 10 students who answer the students correctly in the fastest time will win a prize. And the prize is a $15 credit on your student card towards the purchase of a meal at any on-campus dining location, which I think will be open this fall, fingers crossed. Next, after that, after your skill testing questions, there will be a moderated question and answer session, which will occur in breakout rooms with one speaker and one moderator in each breakout room. And you'll be able to choose which room or rooms you want to visit and join on your own. And then a few minutes before noon at the end, we'll return to the main session for some final remarks. So I'd like to introduce our first presenter, Professor Lilian Chapman. Lilian is an expert in aquatic ecology and conservation, hypoxia and environmental stressors. And she's also been involved for a number of years, for a long time, in the East African Field Studies semester. So thank you for participating in this, Lilian, and I'll turn it over to you.
Thank you, John. I'm just going to share my screen here. And then we will go ahead. Preeti, can you just let me know if everything's A-okay with this screen?
Yeah, I can see everything.
Okay, well, you know, it's really a pleasure to be here today and to share with you a synopsis of my research program. I'm just going to see if I can, there we go. Did you know that over 18,000 species of fishes live in fresh waters, around a quarter of vertebrate diversity? Sadly, though, around a third of these are now threatened with extinction. So in my lab, what we do is we study ecological and evolutionary consequences of environmental stressors for freshwater fishes. And we investigate various stressors, things like hypoxia or low oxygen conditions in the water, thermal stress associated with climate warming and deforestation, turbidity of the water, overfishing, and invasive species. And we do this using a combination of lab and field studies, some of which you see here. Now, we do our research primarily in the country of Uganda in East Africa, where I worked for over 30 years in collaboration with Ugandan scientists and local community members, and where several of my graduate students have carried out their research. Now, we're also engaged on studies that look at the effects of stressors on imperiled Canadian fishes. But what I want to do today is focus on one stressor that we study in East Africa, and that's hypoxia. Why hypoxia? Well, first, hypoxic waters do occur naturally in waters that are characterized by low light levels and low mixing, like these tropical swamps. But the problem we have today is that hypoxic waters are becoming more widespread as influx of waste and fertilizers accelerates eutrophication and pollution, to the point where hypoxia is now considered as an environmental stressor of global concern to fresh and coastal waters. Now, where we work in East Africa, hypoxia or low oxygen is naturally occurring in widespread in dense wetlands like papyrus swamps that you see here. Now, in the papyrus, which average 4 to 5 meters in height, low light levels meeting the waters beneath, low levels of mixing in the waters, and really high rates of organic decomposition produce extreme hypoxia in those dark waters beneath the swamp forest. Now, over the years, we've discovered many fascinating adaptations of fishes to low oxygen stress in these swamps. Let me tell you about one of our focal species. This is a little colorful cichlid, Pseudocretolabris multicolor. It's a maternal mouth-brooding fish. And it's really interesting because some populations of this species actually persist throughout their lives under extremely low levels of oxygen, while other populations are found in well-oxygenated lakes and rivers. And we can compare these. And when we do, we find that P. multicolor from those hypoxic swamps have larger gills, larger heads, probably to accommodate the gills, smaller brains, and smaller young than fish from high oxygen river sites. And when we take those fish and we transfer them alive to our aquatic facility at McGill, and we breed the adults and we raise their young under either low or high oxygen treatments, the young reared under hypoxia have larger gills, larger heads, and smaller brains than their full sibs, their brothers and sisters, reared under high oxygen conditions. Now, this is a good example of phenotypic plasticity, and this might be one mechanism by which fishes could respond, at least some of them, to anthropogenically or human-induced hypoxia and buy some time for evolutionary change. So with that, I'm going to thank you for listening, and I hope I see some of you in a breakout room. Thank you.
Thank you very much, Lauren. It was really, really interesting. And I actually have a few questions, but I'll hold my questions. Okay, so let's move on to our second speaker, Professor Lilian Childress in the Department of Physics. And Lilian is an expert in quantum optics, condensed matter systems, and she's very interested in diamonds and also their defects. So I'm quite interested in listening to this talk. So welcome, Lilian, and thank you for your time today.
Thank you so much, John. Just to check everything is okay, you can see my screen?
Looks great.
Awesome. So this morning, I'd like to tell you a story about spins, photons, and defects in diamond. And let's start with that last part. We work with diamond. And when you think of diamond, you might think of a gemstone, or if you're a chemist or a physicist, maybe you think of a covalently bonded network of carbon atoms arranged in perfect periodic order. But it turns out some very interesting things can happen when you introduce a mistake, that is a defect, into this crystalline structure. Here's one kind of defect we work with in my group called a nitrogen vacancy center, where a nitrogen is next to a missing carbon in the diamond lattice. So what happens when you introduce this type of defect? Well, one thing that can happen is that the diamond changes color. The electrons bound to these defects have characteristic excitation energies associated with specific colors of light. And this nitrogen vacancy defect, well, it makes diamonds turn pink. In fact, all of the colors associated with diamonds come from different defects with different electronic transitions. Even more interestingly, the electrons bound to these defects can have a net magnetic moment, like a little bar magnet associated with the defect site. These tiny quantized magnetic moments are known as spins. And since they can exist in different orientations, they represent a place where you could store information. Moreover, because they're so small, and because they're so well isolated from their environment, they behave quantum mechanically and might one day serve as quantum bits inside a future computing device based on the principles of quantum mechanics. But one of the central questions in realizing such a system is how to get the different spin qubits to interact with each other. One possibility is to send photons back and forth between them. Quantum mechanically, optical information might be encoded in single photons, which are the fundamental unit of electromagnetic energy. But how can you get a single photon to interact with a single spin? Now, I've written out optically active paramagnetic defect center. That's a mouthful. For simplicity, let's just think of this like a single atom. They're really quite analogous. And we're trying to get a single photon to interact with that. How do you do it? Well, you could focus the light down so it's more likely to interact with the atom as it passes, but you can only focus light to the diffraction limit, about a wavelength in diameter or so, and atomic cross sections are smaller than that. In fact, in this geometry, you simply can't have 100% probability of interaction. But if you stick that atom inside an optical cavity such that that focused light gets to bounce back and forth thousands of times before it leaves, then you can, at least in principle, engineer deterministic interactions between a single photon and a single atom, or a single defect center in diamond. And that's one of the problems my lab is trying to solve. In our devices, we build these optical cavities at the tip of an optical fiber. Here's a picture of what that looks like. Then we insert a diamond membrane containing defect centers and assemble the system with translation stages and vibration isolation and lots of other details inside our cryostat, where we're working towards this goal of a coherent interface between single spins and photons. Now, we have a bunch of other interests in our lab, from using these optical cavities to make a better radiation dosimeter, to using spins to measure noise coming from magnetic circuits, to optimizing all of our measurement strategies. But I'm out of time, so let me just thank my group and you for your attention. And I hope to get a chance to chat with some of you more.
Thank you very much, Lilian. Really amazing. You sound to me like a mineralogical physicist. But that's my bias. Thank you very much. That was really great, really interesting. And let's move to our third speaker now, Professor Brendan Johns. in the Department of Psychology who deals with computational models of cognition. He's very interested in big data and also things like language processing and memory retrieval. So over to you, Brendan.
Thank you very much. Oh, does that look okay?
Yes,
right.
Good. Very good. Very good.
Perfect. Thank you. Okay, so yeah, so my research, I'm a cognitive scientist. I'm interested in language and memory, and like many different areas of science these days, I use modern tools from computational science, like big data, machine learning, to sort of develop new kinds of methodologies to examine psychology. So the sort of subfield that I do research in is called computational cognitive science, which actually has a pretty rich history dating back to the 50s and the development of computers. And so the idea is that when us as cognitive scientists, we want to understand kind of the machinations of the mind. In computational cognitive science, we want to understand sort of the algorithms that underlie our ability to do things like speak and remember and make decisions and perception. And so the general framework is that people receive input from the environment that they are embedded in. They process that information in some form, and we produce behavior that's somewhat informative and adaptive to our environments. And so the traditional focus of computational cognitive science has been on examining behavior, highly controlled studies, highly exposed, controlled empirical studies of how humans produce language under different circumstances, which we can then use to modify and construct theories of that behavior. And historically, what has been ignored is the environment that people are embedded in, mostly because there's been technological limitations in terms of what we can access about somebody's history, right? We can't go back and know all the different books that somebody has read or all of the different images that somebody has processed in order to predict the types of how they might operate in a given experimental circumstance. And so my own research uses big data, focusing on language mainly. And so we can take our models of cognition that learn in realistic fashion, how we think humans learn, and we can train them on the same amount of data that a typical or average human might receive. And we can construct new theories about how that input and that just being embedded in a structured environment can impact somebody's behavior on different tasks and in their own life. And so I have a little video here of a model that we use in my own research. It comes actually from a model that was developed in the 80s, from the mathematical models of memory that were developed in the 80s to scale and apply it to learning from a corpus of textbooks, about 10 million words, so it's a rather small corpus as far as these things go, of textbooks from grade 1 through 12. And so what you're going to see here is a neural representation that a model is developing. You're going to see the similarity of these 3 categories, or these 9 words across 3 categories in a 2-dimensional space. And this graph down here is going to show the similarity of the within category objects. And so what you'll see at the beginning of learning is that the neural representation that the model is constructing is pretty random. It hasn't had enough experience to actually develop a coherent representation. But you can see as it's processing more and more language, the actual similarity of the categories are starting to emerge. And you can see that they're starting to cluster together in space. And so that sort of demonstrates that the environment, or the language environment that we are embedded in and learning from, is highly structured and allows us to acquire sort of rich representations of the meaning of those words sort of directly from our experience. And so that's kind of the general framework of the models that we try to develop in my lab. Thank you.
Thank you very much, Brendan. That's really, really interesting. We really appreciate your time. Okay, great. So let's move to our 4th and final speaker, Ms. Odessa Grimald, who graduated from the Department of Psychology and will soon be joining the McGill Faculty of Medicine and Health Sciences. So Odessa, I'm not quite sure what you're going to be talking about, but I'm very much looking forward to it. So over to you.
Awesome, thank you so much. So I'll be talking about veteran sleep, but also a couple of other thoughts on research. And as was said, I am Natasha Odessa Grimard. I go by she, her, they, them. I graduated this past May with a Bachelor's of Science with an honors in psychology. I've been lucky to do a ton of research throughout my undergrad. Currently, I'm with the McGill Comprehensive Health Improvement Program, CHIP for short, under Dr. Stephen Grover and Dr. Ilka Lowenstein, as well as in the Health Psychology Lab under Dr. Barbel Knapper. I would absolutely love to talk with any of you about kind of my research journey and how to get into research, because that was tough. And there's a lot of interesting pieces of that. But what I really want to tell you guys about today is an ongoing research project of mine, which aims to improve veteran sleep and what I've learned so far from it. It all started because this past school year, I completed a senior honors thesis with Dr. Knapper on a project that emerged from her collaboration with CHIP. This project is an online free comprehensive wellness program for veterans and their family members. It's called Mission VAV. If you're part of those groups, absolutely feel free to check it out. It's awesome. I focused my research on a weight loss mission within that broader program, and I also volunteered as a virtual health coach for participants throughout the year. Through this, myself and Max, another health coach, saw that sleep was a really major issue for our participants. With the help of Dr. Grover and Dr. Lowenstein, we set out to build a new mission entirely focused on sleep this late February. Now, at this point in the presentation, I really wish that, like all of the other wonderful speakers that we've had so far, I had beautiful graphs that showed you what had happened, but we're not quite there yet. We're just programming the entire thing, and we're planning on running it for the first time with real humans in late October. Pretty graphs will not come until January at least. But that's okay. I might not have those graphs yet, but this project has taught me plenty of conclusions, and just research in general has. So here's a couple of my favorite ones. First, undergrad research, and I dare say most research, is like 50% hard constant work, but also 50% a series of happy coincidences. I never went into my senior honors thesis on weight loss expecting to come out of it designing a sleep intervention. What got me there was a combination of amazing profs, wonderful colleagues, luck, deep curiosity, and dozens upon dozens of Zoom meetings and emails and hours of reading papers. Second, a research community for you is out there, and you really ought to find it as soon as you possibly can. Prior to all of this, I did cognitive research, a little like the one that Brendan did, which was absolutely amazing and taught me so much. But I always felt a little bit like the odd one out. I was close.
Sorry, Odessa, sorry, your slides aren't changing on the screen.
Odd. Thank you. We are going to see if I can change that. 2 seconds, please. Can you guys see the difference now? Yeah, awesome. Apologies for that glitch, everyone. And so we're back to conclusions. So jumping right back in, I was talking about how a research community for you is out there. So I did cognitive research and it was amazing, but I always felt like the odd one out because it was fascinating to me, but not quite as fascinating as for everyone else in the room. And that kind of convinced me that, hey, maybe I'm just not fascinated enough with research. But I was wrong. Once I joined the Health Psych Lab and CHIP, I realized that I was just as passionate as everyone else in the room, and amazing collaborations have come from that. So it's really worth finding your field and sticking to it, because then it'll just feel like a family forever. And finally, my last conclusion is just follow the dopamine. That's what I did with the sleep intervention, right? There was a little snippet of a subject that caught my attention, and I asked about it, and here we are. So if that happens, or if a conversation in the Zoom breakout room piques your curiosity, Google Scholar is your friend, and your profs are too. Just chat with them about it, and who knows, you might be prying open the doors to a whole new research project. So thank you so much for listening, and a special thanks to all of these amazing humans who've been involved in my undergrad research journey. Thank you.
Thank you very much, Odessa. And I have to say, I concur with everything you say from a professor's point of view. And yeah, I would say just to build on what Odessa said, if you're interested in research, which we hope you are, reach out to a professor who interests you in your own department, or talk to other students in the department to find out what you might get involved in. And also, one last thing, again, following what Odessa said, in research, you have to expect the unexpected.